Hi everyone, my name is Shagher and with my friend Janine we are going to talk about a fascinating way to treat cancer using gold nanoparticles to fight tumors through something called photothermotherapy or PTT. Here's our article that's named that beating cancer in multiple ways using nanogold particles. But today we are going to explain more about using photothermotherapy or PTT to beat the cancer. The basic idea is pretty cool. We use light to heat up gold nanoparticles, and that heat is used to kill cancer cells in a very targeted and controlled way. Think of it like having a microscopic heater that turns on only at the cancer site. So, photothermotherapy is a minimally invasive technique where the light energy is converted into the localized heat and then leading that to the selective destruction of cancer cells. Like here, I explain it with the photos. And so gold nanoparticles are really ideal agents for us for this situation because of their unique optical and physiochemical properties, especially their ability to absorb light and efficiently convert it to the heat. So here is the question, a really important question. Why gold and why gold nanoparticle? What makes gold nanoparticles so special here? They have a unique property called surface plasmon resonance here. And when a gold nanoparticle are exposed to near infrared light, the NIR, typically the wavelength is typically between 650 and 900 nanometers, then the free electrons on their surface oscillate in a resonance with the incident light and start to vibrate and turn that light into the heat. These oscillations causes two things: intense absorption of the light energy and rapid conversion of the energy into the localized heat. So in this diagram, I show that the wavelength and the NIR tissue transmission rate, and then the electrons, the free electrons oscillate and vibrate and turn that light to the heat. Uh, as I explained, I can explain more about this unique the properties of the gold nanoparticle. They can synthesize into the different shapes, including nanorods, nanoshells, and nanocaves. So they can absorb the exactly the right kind of light, which allows us to tune their optical absorption precisely into the NIR region. So they can take that the right kind of light in the NIR regions. But another question here comes, why NIR range? So because of their NIR light penetrates several centimeters into biological tissues with minimal absorption by water or hemoglobin. So that enables the non-invasive access to deep tumors. That means that that's the range where the light goes deep into the body without being absorbed by blood or water. So you can imagine this like using a TV remote that sends a signal that passes through the air and only activates the specific devicer or the receiver. Here, the NR light passes through the tissue and only activates the gold nanoparticles deep inside the tumor or tumor tissues. But uh, the other hand, we can say that the heating or converting light into the heat is not enough on their own. We need to be sure or make sure that the heat and this process goes only uh, to the tissues and the cancer tissues. On that's where the targeting comes in, the targeting ligands. To achieve this, we can say that the gold nanoparticles are really functionalized, meaning their surfaces are coated with targeting ligands, such as antibodies, for example, anti-EGFR or anti-HER2, or peptides or small molecules that selectively bind to the cancer-specific receptors. So we can attach the antibodies or special molecules uh, to the surface of gold nanoparticles once that they recognize cancer cell receptors like EGFR or HER2. Plus, tumors have abnormal and leaky blood vessels, a phenomenon called the enhanced permeability and retention, the EPR effect. So, nanoparticles are naturally accumulate more in tumors than in normal tissues. Once the particles are at the tumor site, a low power NIR laser is applied externally. That's how we can kill the cancer cells. The gold nanoparticle absorb the light, heat up, actually convert it into the heat, and kill the cancer cell and cause localized hyperthermia, either by triggering apoptosis, a kind of cell suicide, that the meal heating, we need the meal heating like 42 to 45 degrees, or necrosis, that we are breaking the cell membrane and we need a stronger heating more than 50 degrees. So here the key advantage here is that the healthy tissue around the tumor stays unharmed. So that's a good news, as the heat generation is localized strictly around the nanoparticles. We can imagining like using a magnifying glass to focus on sunlight and burn a single spot on a paper, that we are in our childhood do that, for example. Except here we are happening the all things happening inside the tumor or tumor tissues on the nanometer scale. So as a result, I gave you two diagrams. The MR-guided tumor temperature mapping, showing the increase in tumor temperature during the laser exposure. And the other one is tumor volume reduction after photothermal therapy, and actually after a single photothermal treatment, demonstrating the therapeutic effect that for example, after the 12 days, we have 0% of the tumor volume. And the other can be explained by Jenny.
Yeah, now that we know how it works, I will continue with the results obtained from the experiments. And in one experiment, gold nanoshells were injected in mice with breast tumors. And after near infrared laser exposure, the temperatures inside the tumor rose over 37 degrees, while the control group saw minimal heat increase. So the tumors in the treated mice were completely destroyed after a single therapy session, and the animals remained tumor-free over 90 days. And similar results were seen with gold nanorods, and they provided even a better heat generation. In one study, over 57% of tumors disappeared in one treatment with locally injected nanorods. Even systemic injections worked well thanks to pegylation. This is a coating that increases circulation time and minimizes immune detection. Yes, beyond that, gold nanoparticles aren't just therapeutic, they're also diagnostic. So they act as contrast agents in CT, MRI, and photoacoustic imaging, which means we can see where the particles accumulate and monitor tumor shrinkage in real time. And this combination, imaging and therapy, is called theranostics, and it represents a major step toward personalized precision medicine. So you can imagine, a single injection allows us to both find the tumor and destroy it at the same time, all with a simple light source and no harsh drugs. Yeah, now we already come to our conclusion. Our question was how gold nanoparticles greatly enhance the efficiency of photothermal therapy. And yes, they do it by precisely converting light into heat, by targeting only tumor cells, by enabling real-time imaging, and causing minimal side effects. So all in all, they are safe, they are tunable, and incredibly effective. And with more research and clinical trials underway, gold nanoparticles could soon become a cornerstone of non-invasive cancer treatment. And yes, thank you for listening, and we hope that you found our video and the informations as fascinating as we did. And yeah, we see you soon.